Do you know what is nutrigenomics? Nutritional genomics or Nutrigenomics is a study of the interaction of nutrition and genes, or the effect of nutrition and other bioactive food components on the expression of genetic material. Human genetic variation can predict how the body will respond to different nutrients. Genes have long to be assumed as the hardwired blueprints of our bodies. Although genes play a critical role in predetermining certain factors about our health, the emergence of epigenetics nowadays has shown us that our environment can modify genes. Because what we consume is an essential aspect of our internal environment, nutrition strongly influences whether genes are activated or deactivated. Note that some individuals will respond positively to some diets more than others who may get no or an adverse effect. This response is based on the principle of nutrigenomics. Eating a personalized diet can eventually impact genes that will be expressed and simultaneously humans' overall health. Here, let's see some of the examples of the adverse effect.
Nutrigenomics is the study of how naturally occurring chemical in food alter the molecular expression of genetic information in each individual. When a gene is activated or expressed, a protein is produced. Gene expression pattern produce a phenotype which represents the physical characteristic or observable trait of an organism. Gene alone do not necessarily produce phenotype or trait. Phenotypic expression is influenced by nutrition. Nutrigenetic is the study of how individual humans respond to food based on their genetic makeup and how genetic susceptibility to disease can be manifested or mitigated through nutritional consumption. Nutrigenetic focuses on the individual and identify the type of diet a certain person should follow based on his or her gene.
The emergence of nutrigenomics in this era is scientists have been able to identify the rare genetic variants or the single nucleotide polymorphisms that affect the lipid profiles and the risk of developing the dyslipidemia. These SNPs have been linked to dyslipidemia through comprehensive genome-wide association studies. Our understanding of lipid metabolism and customized treatments has been completely transformed by nutrigenomics. This result emphasized the need for personalized management strategies for dyslipidemia and the significance of genetic variables in determining the individual vulnerability to the illness, which hold the considerable potential for enhancing cardiovascular health outcomes, highlight the need of incorporating nutrigenomics into clinical practice and public health initiatives. As we enter this new era of telemedicine, nutrigenomics will continue to provide insightful information on dyslipidemia.
Different persons have different polymorphisms, and the most common type of variation, known as SNPs, change metabolic response to diet and influence the risk pattern of disease. With the presence of nutrigenomics, the nutritionist can provide dietary recommendation that meets patient personal nutritional and health needs, as well as prevent nutrition-related chronic diseases based on an analysis of genetic makeup. A personalized nutrition plan can help them achieve dietary and physical activity targets that are more on track with what works for their body. Nutrigenomics can increase awareness of risk of certain conditions by increasing people's understanding of how nutrition influences metabolic pathway and alters homeostatic regulation in our bodies. Nutrigenomics also can improve health quality and quantity of life. The person's vulnerability to developing the diet-associated disorders can be quantified, and personalized nutritional guidance can subsequently be provided to that specific individual. Unfortunately, there are some misleading claims about nutrigenomics. Firstly, genetic testing kit is raised concern by providing incorrect and unclear forecasts regarding health risks. Furthermore, the test result may include suggestion for quite expensive dietary supplements, even there are other cheap supplements with similar benefits. Secondly, according to one company that advertises for consumer genetic testing, they claim that people's DNA has a big part in defining how their body reacts with food, impacting preferences, sensitivities, and metabolism. It is incorrect because they make no recommendation and only promise genetically personalized diet program.
The potential role of nutrigenomic field in managing the dyslipidemia is we need to control total fat intake, and it should be consumed 25 to 35% of total energy intake. And for the saturated fatty acid, can only consume 7 to 10% of total energy intake. Reduce saturated fatty acid intake, lower dietary cholesterol intake, since saturated fatty acid and cholesterol often coexist in the same food. When the saturated fatty acid replaced with the polyunsaturated fatty acid, there is consistent data that cardiovascular event and coronary mortality are reduced. Trans fatty acid intake should be kept at less than 1% of total energy. Prospective cohort studies show that trans fatty acid also associated with an increase in the incidence of diabetes. It was estimated that substituting 2% of energy from trans fat with saturated fat, MUFA and PUFA will reduce cardiovascular risk by 70%, 31% and 24% respectively. Monounsaturated fatty acid intake should be taken 12 to 50% of total energy intake. There is convincing evidence that replacing carbohydrate with MUFA increases HDL cholesterol concentration, and that replacing saturated fatty acid with MUFA reduces LDL cholesterol and the total to HDL cholesterol ratio. Several researchers have shown that replacement of MUFA enhances a rise in the concentration of HDL cholesterol and decreases triglyceride. It also lowers the susceptibility of LDL cholesterol to oxidation. Lastly, using lipid-lowering drug LDL-C reduction with statin treatment remain the cornerstone of the lipid-lowering therapy to reduce risk of cardiovascular disease because of the consistent result of numerous randomized primary and secondary prevention clinical trials. Depending on the treatment level of LDL-C level achieved, lipid-modifying agent can slow the progression or even promote regression of coronary atherosclerotic plaque. To prevent the disease, firstly, we eat food that are high in fiber, especially solid fiber. Second is choose food low in saturated fat, trans fat, sodium, and added sugar. Besides that, choose white meat instead of red meat. Next is limit to add yolk per week. Then is quit smoking. Furthermore, is limit alcohol intake. Besides that, choose low fat or skim milk. Moreover, limit food high in saturated fat. Next is exercise at least 150 minutes a week of moderate aerobic or 75 minutes a week of vigorous aerobic exercise. Lastly is do screening for people with risk factor of dyslipidemia regularly.
The LDLR and ApoB genes are the most commonly discussed genes that are associated with dyslipidemia. First, LDLR gene. The receptor-mediated endocytosis process uses a cell surface glycoprotein called low-density lipoprotein receptor (LDLR) to bind to and take up plasma LDL particles from the blood circulation. It significantly promotes cellular cholesterol homeostasis. Plasma LDL levels may significantly increase as a result of polymorphic variants in the LDLR gene, raising the risk of atherosclerosis and coronary heart disease. FH is a type of extremely high blood cholesterol caused by mutations in the LDLR gene. Some of these genetic alterations decrease the amount of LDL receptors produced by cells. Second, ApoB gene. ApoB is the main protein component found in lipoproteins, which are the carriers of lipids in human blood. ApoB is a key that attaches to LDLR on the cell, causing it to take up lipid-rich lipoproteins. Faulty ApoB, similar to LDLR mutations, inhibits the removal of LDL from the blood, causing the development of plaque and atherosclerosis. Each mutation that results in this disorder alters a single protein building block of amino acids in a crucial area of a polylipoprotein B100, while a polylipoprotein B48 is normal. The modified protein stops LDLs from successfully binding to their receptors on cell surfaces. Therefore, less LDLs eliminated from the blood and blood cholesterol levels are significantly elevated.
ApoB gene variants can change secondary structure of the human ApoB and the LDL particle size compared to wild-type LDL particle. Such studies of ApoB variants may provide useful information to understand their role in the LDL oxidation, dyslipidemia, and atherosclerosis progression. Studies show strong connection between the SPA1 SNP and polymorphism of the ApoB gene, which del variant of the latter is also consistently associated with serum LDL elevation. According to AUS 2020, in a Brazilian sample, the haplotype formed by the del and T allele shows strong association with a mixed lipid profile of higher risk for coronary heart disease. It has been proposed that the 3 amino acid missing in the del variant of ApoB can alter the hydrophobicity of the signal peptide and impair translocation of the newly synthesized ApoB from the cytoplasm to the endoplasmic reticulum, affecting the secretion of ApoB as VLDL from hepatocytes. In familial hypercholesterolemia patient, LDL receptor function is reduced or absent, leading to reduced LDL removal from circulation. According to Melancholy 2019, violent loss of exon 9 of LDLR gene in the reported case may result in impaired release of LDL from the LDL receptor in the lysosome, thus potentially interfering with recycling of the LDLR from the lysosome back to the cell surface. The EGF precursor homology domain also helps in binding of LDL on the cell. However, homozygous deletion of exon 9 of the LDLR gene may result in malfunction of the EGF precursor homology domain and might disturb normal orientation of the ligand binding domain of the LDL receptor and resulting in loss of LDL binding to the LDL receptor.
What is the status of acceptance of nutrigenomics in Malaysia? Based on an article entitled Predictors of Stakeholders' Intention to Adopt Nutrigenomics from the National Library of Medicine, this study found that the intention of Malaysian stakeholders to adopt nutrigenomics is characterized by a complex decision-making process involving interrelated factors. The perceived benefit-perceived risk balance is a crucial element in deciding whether or not to support nutrigenomics. When stakeholders perceive that the benefits of nutrigenomics exceed the risk, the perceived benefits are significant in influencing and predicting their intention to adopt nutrigenomics. Researchers and practitioners should note that crucial role of perceived benefits as a direct predictor and mediator for all other predictors. Therefore, it is expected that increased exposure to the beneficial aspects of nutrigenomics and the process of involved will influence the beneficial perceptions and thus support the applications. The influence of other factors, for example, trust in key players and religiosity, should also be considered when devising an appropriate technology acceptance strategy. Although the overall findings showed that Malaysian stakeholders were enthusiastic about nutrigenomics, they were also wary of implementing it.
An increasing number of people carrying diseases, especially dyslipidemia, are concerned in Malaysia. Personalized nutrition should replace the one-size-fits-all approach which is not getting to grips with the health risks currently affecting Malaysian people. Developing optimal diet for an individual based on their genetic makeup has the potential to drive down rates of common diseases in Malaysia. Hope that with more advanced technology along with more smart ways to be healthy, will decrease the amount of people with disease and to maintain the healthy lifestyle that already exists.